Senior Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is The Transition to Automated Library Prep for Enhanced Pathogen Surveillance Workflows, and is sponsored by SPT LabTech. Our speakers today are Krystal Sheridan, the Sequencing Laboratory Supervisor, Pathogen Genomics at the Translational Genomics Research Institute, Eleanor Matrullo, the Automation Program Manager at Watchmaker Genomics, and Melissa Netwall, a Field Application Scientist at SPT LabTech. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. And if you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. And with that, I'll turn it over to Krystal. Krystal?
Hi, thank you very much for that introduction. My name is Krystal Sheridan. I work for TGen, or the Translational Genomics Research Institute, or TGen North, as we're called. We are the Pathogen Genomics and Microbiome Center for the larger TGen organization. I manage a sequencing laboratory, and I'll be talking to you today about our group-based strep surveillance program in Arizona. And how we've kind of optimized that workflow. So let's see if I can get through here. Okay, so as was already said, the title of the presentation today, The Transition to Automated Library Prep for Enhanced Pathogen Surveillance Workflows. And all right, a little about TGen to start. We are located in Flagstaff, Arizona, which is just south of the Grand Canyon, if you're familiar with that. We were established in 2007, and I've been there for about 9 years. We are a nonprofit organization, research institute, and like I said before, we focus on pathogens and microbiome. Little information here. Oh yeah, let me get my arrow. There we go. This little symbol here shows that we were first responders in terms of genomic response for COVID-19, and we are an integral part of the Flagstaff STEM City community. And we are also an affiliate of City of Hope Hospitals. Sorry, I went a little too fast there. Our mission at TGen North is to advance human health through next generation microbial science. And we have this motto here that is, we have the ability, therefore we have the responsibility. And this is kind of some of our lab folks doing some hard work there. Our focus areas are bacterial and viral pathogens. We look at antibiotic resistance. Over here you can see that we have a haboob. We have these fun dust clouds in Arizona that bring with them valley fever, or coccidioides, which is a fungal pathogen. We have enteric disease surveillance programs, genome tracker with the FDA, wastewater-based epidemiology, so looking at different pathogens in our wastewater. And lastly, on this slide, we have vector-borne and zoonotic diseases like West Nile virus and mosquitoes. Today, we'll be looking a little bit at healthcare epidemiology, and another one of our focuses are HAIs, or healthcare-associated infections. We look at these a lot, especially in special populations like detention centers or underserved populations like tribal communities, and we have a really healthy partnership with regional and border health. So our star organism today is the Streptococcus pyogenes bacteria. This is group A strep. This is a gram-positive bacterium, which means it needs a little extra lysis when you're extracting DNA. It's beta hemolytic, so you have a culture that's pretty identifiable because it kind of eats the blood around it. The M gene is what kind of gives it the virulence. It's how we tell whether one strain is more virulent than another. Person-to-person transmission is how this is spread by direct contact with discharges from the nose and throat of infected people, or by contact with infected wounds or sores on the skin. The risk of spreading the infection is highest when a person is ill, like if you have strep throat or an infected wound, but you can also carry the bacteria without having any symptoms at all. So asymptomatic carriers are possible, but you're less likely to spread in this case. Mild to severe illness is possible. There's a whole range of fun stuff. And invasive infections are tracked by the CDC's Active Bacterial Core, or ABC, surveillance program, but just invasive infections. So we also look at invasive group A strep. And invasive in this case means that the bacteria can invade parts of the body that are normally sterile sites. So this will result in severe disease outcomes and high mortality rates, like getting into your cerebral spinal fluid or your blood. Like I said before, a range of illness is possible here. Mild disease from pharyngitis or strep throat to scarlet fever. And this image here kind of showing what it looks like when you have strep throat. And some severe diseases caused by this bacteria are necrotizing fasciitis, also known as like flesh-eating bacteria, or toxic shock syndrome, or sepsis. And so I'll show you an image of the necrotizing fasciitis, but if you don't really have a stomach for kind of graphic medical image, please look away for this moment. And so here we have an example of necrotizing fasciitis and how that can affect the body. And you can see that it is pretty severe and not something that you really want. Okay, the image is gone if you didn't like that. Moving on. I'll talk to you a little about our experience with group A strep in Arizona. In 2015, we found a highly virulent group A strep. We did whole genome sequencing analysis of isolates during 2016 to 2018, which was performed on clinical samples collected throughout the state. The results showed an emergence and increase of uncommon M types, so like this M59 here. And we found a distinct clone that were separated by just a few SNPs, 0 to 4 SNPs. And this was genomically supporting the presence of an ongoing outbreak at the time. And this occurred in northern Arizona. So this here is kind of a look at our hospital emergency center. And in terms of M59 tracing, a little more of the background for the paper that I just showed in the last slide. This is a table from that paper showing some of the clinical presentations or demographics. So some risk factors: injury, alcohol abuse, homelessness, local jail term, and kind of how they were presenting: necrotizing fasciitis, 5 cases, sepsis, 9, cellulitis, 7. And then we have some age information and race information here as well. This would be our phylogenetic tree that was drawn out after we looked at isolates throughout, historical isolates back through, I think it was 2009, and then all the way to 2015. And here in blue are the AZ samples. Sorry if you just saw a bunch of little arrows. I didn't find the right place to click for a second. And we can see that these are on the same branch of the tree. They're kind of collective on the same nodes. And you can see that they are caused by a single clone that's also closely related to this New Mexico isolate. And we do also have one little outlier here, patient M. So if you look at the arrow, that one was kind of an anomaly, but all the rest of our AZ samples were kind of gathered in the same area of the tree. Not only were these cases genomically related, but epidemiological investigations confirmed that the patients were associated with the same jail setting. And there was a fight that was reportedly broke out at the jail. And we think that this was the transmission pathway for these group A strep infections. And most of the case patients were Native Americans, 3 of whom actually passed away. And risk factors for these folks included homelessness, alcoholism, incarceration, and IV drug use. And so given that we did have a pretty big impact in figuring out kind of what happened there in that group A strep case with the M59 type, a little bit later in 2019, the Arizona Department of Health Services requested all IGAS isolates be submitted to the Arizona State Laboratory, which are then routed to TGen, where I'm at, for genomic analysis. So we now perform routine genomic analysis on group A strep samples to detect emerging strains, identify outbreaks, and create an informed public health response. So it's really important to understand the risk factors, to have rapid outbreak response, and to implement prevention measures when we know something's going on. It's a little chart here that shows from 2009 to 2022. This data is from AZDHS, and we have sequenced some of these isolates as well. And this just kind of shows you the increase in reported cases of group A strep in Arizona. The M59 outbreak happening in 2015, you can kind of see that's where it starts to really, the rates start to go up after that. And this is just an example of the rates themselves graphed out for you per 100,000 population. So conducting group A strep surveillance at TGen North. Dive into the details of what my lab does. We are called the Hive Laboratory, and it is not an acronym, and I didn't name it, but I like it. So that's who we are. This is my technician, Bella, and she pretty much is the workhorse for group A strep and processes all of these samples. This here is a plate culture of group A strep. This is what gets sent to us from Arizona Department of Health. So they culture, confirm, and then send to us for sequencing. A little about our laboratory, we do high throughput sample processing and next generation sequencing. We've sequenced greater than 2,000 IGAS isolates for the Arizona Surveillance Project. And we support a lot of the larger projects at TGen North, so specimen processing, culturing, extraction, library construction. We do some automation and sequencing. We have Illumina and Oxford instruments. Our workflow for group A strep starts with a bacterial culture. Sorry, that just added all the little QC steps. So as you can see here, plated out in a Petri dish. We then make glycerol stocks so we can go back to them if we need them. And we extract DNA with a QIAGEN kit. We QC the DNA with a genomic tape station, normalize the DNA for input into a watchmaker library prep. We do the whole genome sequencing library construction. Then we pool libraries after we QC those libraries, and we do equimolar pooling for that. And then we sequence. So down here in this image, we have the Firefly and an Integra Assist. Those are automation instruments that we utilize. And then we have a NextSeq 550 that typically is used for sequencing. I want to focus on some kind of problem areas that we've identified, things that we could do better, and talk to you a little bit about that. So some of the time-consuming and error-prone steps that we've found happen in the normalization step, especially when it comes to arraying large sample sets. So making sure they're all in the right place, making sure that we're not cross-contaminating the plate, normalizing samples with variable dilution requirements. If you work in a lab and you do wet bench work, you may know how miserable this can be to put different volumes into every well of hundreds of sample wells. And it can become inconsistent based on how well your pipettes are calibrated, things like that. And for library construction, we have bead cleanups and size selection can be in the vein of a wet bench person's existence. Your technicians sometimes do hate this. You try to get clean product and it's really difficult. Pipette mixing with viscous liquid classes can be difficult at times, and maybe you don't always get everything mixed up. Sample transfers can cause cross-contamination, and sometimes we have seen, an accidental plate flip or something like that. And then reaction setup, maybe you forget one piece of the one element of the master mix. Mistakes do happen. In pooling libraries, we do see some pooling errors occasionally, a missed sample or a pipette error where the volume was too low for the pipette. Or something of that nature. Uneven sequencing results result from those pooling errors. We get insufficient coverage. And sometimes there's an index misassignment. So if the wrong index was put into a sample and then assigned to it. How can we improve this? I think we can standardize our procedures and training, which we have worked hard on doing at TGen North. Utilize controls to track some of these issues, which we also have incorporated. We also have incorporated calibrating pipettes more often. But something we really wanted to do is make some time to test multiple library prep methods and reagents to see, if we can improve our results. What I find important for good library prep reagents are good quality fragmentation enzymes, I really want to have consistent results and avoid the need for a double-sided size selection because you lose a lot of product when you do that, or lose a lot of your DNA. So it'd be really nice to have a good quality fragmentation enzyme that isn't too temperature sensitive so that you end up with, consistent sizing. Ligation and conversion efficiency, so the ligase enzyme and how well everything converts to a ligated library that is sequenceable. Adapter ligated library, I should say. And adapter dimer, don't want it. This stuff is not fun for sequencing. It takes up sequencing runs, especially for short read sequencers. So we really try to avoid generating this by not cycling too much or finding reagents that actually work better in not creating it. Want an input quantity range. At TGen North, we do have variable sample types, so we need something that can do somewhat of a low input and also not be too inhibited by a higher input as well. An easy to follow protocol, essential, right? We want to be able to train easily and be able to get through it without too much trouble. I want something that takes at least, like less than a day, maybe something reasonable, just a couple hours would be great. We want good support, technical, sales, automation support, all of this, so that if we have issues, we have someone to turn to. And at the end of it, we need a certain amount to sequence, so at least like 2 to 4 nanomolar. And we want a kit that's going to produce libraries and sequencing results that have minimal sequencing artifacts and bias. So sometimes you see a start bias from certain kinds of cut enzymes that choose specific areas, or GC content issues where organisms are not covered the same, which especially for microbial sequencing is important because we have a variety of different organisms. What we did was evaluated 7 kits and ultimately decided to use Watchmaker Genomics library prep with fragmentation kit. This is a little picture of it right there. We actually evaluated 6 of these at the Collaborative Sequencing Center at TGen headquarters. And I compared the additional kit that TGen North was using when I moved here to everything else, and still kind of decided that Watchmaker was the best fit for me. What did I like about this? Kind of everything I just said, but they did have a robust fragmentation step that was tunable, which I really love, and produced good results. You don't need an extra size selection, so no sample loss there. There was a fast and easy workflow. So one of our technicians calls herself a Watchmaker fangirl, so I know that they really like it. It only takes 130 minutes to finish the entire thing. Low input requirements of 100 nanograms work easily and reliably. You only need 4 cycles of amplification to achieve high yields, like 30 to 50 nanomolar post-PCR product. And we've had success down to 4 nanograms of input. There is a PCR-free option, which also works with 100 nanogram input. And there's an efficient ligation and amplification enzymes as well. So we are getting great conversion rates. And last but not least, excellent scientific support team. Automation support is provided. They help you make protocols on whatever you've got. And they are really helpful on the science side of understanding what your needs. Oh, not last but not least, consistent results. So does well with a range of GC content, no sample fallouts. We are just very happy with the results that we're getting here. Here's an example of some libraries that we generated with this kit. This is a D5000 tape station assay for post-PCR products. You can see here, it kind of looks like an electrophoretic image. You can see that it's pretty even. And then if you look over at these individual sample images here, these traces are fairly even across the board in terms of amplitude and in terms of their range. So really happy here. These last 2 at the end are negative controls, so nice and clean, no adapter dimer. That's what we like to see. In terms of conversion efficiency, up here is the equation that we use, which makes no assumption of sample loss through the process. Some clearly does happen in size-selected libraries, which in these experiments, these were done at the Collaborative Sequencing Center using PCR-free workflows. And these ones we used inputs based on supplier recommendations. So the different colors up here kind of indicate those different inputs in nanograms. 100 nanograms was the lower side input for enzymatic kits, and this one here is the watchmaker. And it performed very well compared to the other ones, especially considering that it had a lower input to begin with. So we really like that. And also note that this blue one all the way on the left-hand side, we believe is an anomaly, like an error. So not really that actual conversion efficiency. And in sonication, if you look at the y-axis, it is quite a bit lower in terms of the conversion efficiency, like scale from 2 to 4 rather than in the 20 to 30% range. So you lose a lot when you're doing sonication. And these were size-selected libraries. Conversion efficiency calculations in terms of PCR amplified libraries, we did 4 cycles. Use this equation right here. Our input was 100 nanograms, and we eluted in 40 microliters. I'm seeing 70 to 80%, which is wonderful. It might be even higher actually, because I do dilute it a little bit through the process. Okay, so now that I've kind of talked about our background in terms of choosing which library prep reagents we wanted to use, I'm gonna put a check mark by that and move into another method to enhance the workflow, which is incorporating automation. Now, why would we want to automate? Ultimately, it saves money and time to be fast and reliable. You don't have to go back to things, and you get things done to standardize TATs. So if someone's asking you how long it's going to take you to do something, you can pretty much guarantee it when you have automation. It also enables your staff to focus on complex tasks that the robot can't do, so you can free up their hands and get them doing something more important. It helps to prevent cross-contamination, reduce human error. You can track your protocols in the software. Easy to optimize and maintain, depending on your instrument, probably. Consistent yields, and you get reliable sequencing results, which inevitably reduces your sequencing costs. So looking at how we decided to select which automation instrument we were going to purchase, we looked at lots of different companies, Agilent, Epinorth, Hamilton, all the ones that you know and love. And there's something good. There's pros and cons for all of them. So for me, I'm looking for something that's has a good price tag, has a good lab footprint, not too big because we don't have a lot of space. I want software that's easy to use and easy to train everybody, a good support team, protocol development help, and flexibility in terms of how many samples it can do. These are examples of other instruments that we looked at, kind of bigger, some good walk-away capability for some of them, but the cost is the bench space and money. Ultimately, I really liked the Firefly, and this is the image of that instrument here that SPT LabTech makes. It's got an affordable cost, about half the cost of most of the other options that I looked at. The lab footprint is by far the smallest that I saw. The use of vertical space in the instrument is a really cool way to overcome the issue of bench space. The software is very user-friendly. It's cloud-enabled, so you can share protocols, and you can use it from any PC that you have. So support-wise, great experience, on-site training included with the purchase, and I really love their NGS and automation team. I had a great time, and I feel like we're all friends now, so that's cool. Protocol development, automation specialist assistance. So it's easy to develop and optimize your own protocols, but also they will help you, and especially if you're using Watchmaker or another reagent provider that helps you with automation, it's nice you'll have like a whole team on your side. So flexibility on this, and it's easy to mix and match protocols, components. You can change labware without it just breaking the whole program, which I have seen in other instruments, and you can adapt it to different batch sizes. The head on it is 96 or 384. You can also use strip tubes to be able to do smaller sample sets, which is really nice. A lot of other options have closed systems that make it difficult to edit or develop protocols, and they charge per protocol a lot of the time, which was way too rigid for our needs. I think that this system provides modifiable and optimizable workflows that you can adapt to different batch sizes and liquid classes. Some other bonus features, as I said, 384, 96, or columns of tips are capable on this instrument. Positive displacement dispense heads. When you dispense your master mix, it's liquid class agnostic. So you can just do it super fast. You don't have to put in any crazy settings for how it mixes or blowout speeds or any of that. In this case, it just shoots it out there and moves the deck below it. So it's really nice. It's like 30 seconds, I think, to fill a plate. The deck moves around underneath the tips, and then user access control is provided, so it's passcode protected, and you can protect your protocols depending on who's using it. This is what the software kind of looks like, and I included a link, and we'll share the slides later if you want to take a look at that. So this area is where we use the Firefly. We use it for our whole genome sequencing library construction, and we have programmed it for the watchmaker workflow. It takes about 1.5 hours on the Firefly, not including thermal cycling. You can use the Firefly to normalize and pool. And I think that automating library prep kind of gives you that guarantee of consistency. And like I said, frees up other hands. It's like having another person in the lab. You can see here, these are sequencing results. So the percent lane on the y-axis and our different group A strep samples along the x-axis. You can see that the coverage is fairly even, especially in terms if you were to compare this to manual pipetting. And our sequencing depth for COVID-19 is also shown here, which this is actually a really nice and even pool. I'm going to move into the analysis sector now that I've kind of talked about my whole journey through optimizing our group A strep workflow. So just take a couple minutes to talk to you about TGen Epi tools, which is something that our bioinformaticians have been working on. It's a new tool to make epidemiology more accessible and actionable. Democratization of sequencing requires a democratization of genomic analysis. If you have all the data, but you can't do anything with it, what's the use, right? TGen Epi Tools provides public health epidemiologists an easy-to-use platform for genomic analysis. You can draw phylogenetic trees in real time, and user-selected subsets of data can be visualized. Any available sample metadata can be combined with the tree to find clusters, trends, relationships. This is an example of the first version of it that we had. And so you can see that there are different like visualization options. You can change the sizes, branch scales, choose different M types for group A strep. And this is kind of what the image looked like. In 2.0, which is how it looks now, it's a little bit prettier, a little cleaner, and there's a bit more options. This is an example of a SARS-CoV-2 phylogenetic tree. And I'm not going to go through all the details here, but if you want details, there is a paper that we have there. This is just an example of how there are different tree styles that you can choose from. You can link metadata. I love this part, and I think epidemiologists can really appreciate this. So you can select and see your metadata down here in the table, and you can select which pieces you want to actually be visualized on the tree. So if you want collection dates or where it's from, things like that, you can go ahead and select those things in the menu and append metadata. Clusters are automatically detected, and you can choose your sample, your size of cluster and distance of cluster that you prefer. And I think this is my second to last slide. This is just an example of our dashboard. So you can follow this link down here that goes to our group A strep tracker. Right here, we have 2019 to 2023 M-type data for group A strep. And this is kind of something our epidemiologist generates and provides to the public in order to see what M-types are out there right now. So the different colors on the right-hand side show you those. And basically M49 in the dark blue has been dominating the strains so far. And these M-types give us ways to react and respond in the community. The Southwest Pathogen Intelligence Center is kind of the larger part at TGen that collaborates with local, national, international partners to support clinical, tribal, and public health. You can follow this QR code and it'll take you to our website. This is where you'll find TGen EpiTools when it becomes available. It's not currently publicly available, but will be soon. And then you can also find our dashboards. So we have wastewater, viral pathogen tracking dashboards. We have COVID dashboards, and our group A strep, which it was the first Arizona statewide genomic surveillance project for group A strep. And so go ahead and check those out. And just wanted to let you guys know that this is also an early warning system for outbreaks and how we understand the evolution of these microbes. And my last slide, thank you so much to everybody at GenomeWeb, SPT LabTech, Watchmaker, the CSC, our funder NARBA, the Department of Health Arizona, and then all of my TGen folks here. Thank you so much. I will now pass it to Eleanor Matrillo from Watchmaker Genomics.
Thanks, Krystal. All right, so I am Eleanor Matrillo, the Automation Program Manager here at Watchmaker Genomics. And as Krystal mentioned, her group is leveraging our Watchmaker DNA Library Prep Kit with fragmentation in their pathogen surveillance workflows. So we figured we'd take the opportunity to give a very brief overview of Watchmaker and what we do. So we're focused on building precision tools for the life sciences. We're based out of Boulder, Colorado, and we have a second facility in Cape Town, South Africa. So our current focus area is on improving quality of reagents for high stringency molecular biology applications where data quality is critical. And in an ideal world, the data output would be a perfect representation of the information that's carried within a biological sample, but oftentimes this can be challenging. In reality, all biological information is read, written, and maintained by enzymes, and capturing a sample's information really requires enzymatic manipulation at multiple points throughout the workflow. Unfortunately, many times these processes can be quite inefficient and can also result in data corruption. So as we see it, quite simply put, if you can improve the enzymes, you can improve the quality of data. So enzymes are really at the core of Watchmaker's precision product development platform. We leverage our expertise in the areas of protein engineering, computational biology, and high stringency enzyme production to purpose-build enzymes for specific NGS and molecular diagnostic applications, the workflows for which are really developed in parallel. So we use the application's needs to define the enzyme performance requirements, and this fuels iterative enzyme application code development to deliver tailored solutions. So today we'll use DNA library prep really as an example of where there was a technology gap for which we've been able to develop a highly scalable solution that provides superior sequence accuracy. And as many of us know, sonication workflows for DNA library prep are really the gold standard for unbiased, robust DNA fragmentation, but it doesn't necessarily scale very well and it can require quite costly consumables. So sonication workflows also have issues with template damage and loss of material that can impact results, especially for those low input samples. So enzymatic fragmentation is an attractive alternative that alleviates the monetary or barrier to entry and sample processing bottlenecks. But enzymatic fragmentation-based chemistries also have their own limitations with respect to sequence accuracy and difficulty processing a wide range of inputs with a single unified protocol. So our goal was really to develop a solution that harnesses scalability benefits of enzymatic fragmentation while delivering sequencing accuracy that's on par with the gold standard sonication approach. So we developed the Watchmaker DNA Library Prep Kit with fragmentation, and shown here is our total workflow. The kit components are at the top of the figure in purple that are then in some cases combined into master mixes and ultimately added to the sample. So our workflow incorporates an integrated fragmentation, an end repair and A-tailing step that's then followed by a same tube high efficiency ligation reaction in which the ligation master mix is actually added as a single reagent addition. And that makes this step a lot more automation friendly. PCR-free workflows can be finished within an hour and a half, while PCR-based workflows may require up to an additional 45 minutes, depending on the cycling parameters that are selected, plus then a final spry bead cleanup. So our kit supports a wide range of DNA inputs from 500 nanograms to below 1 nanogram with consistent conversion rates and highly tunable fragmentation, really for all input amounts, just by adjusting the fragmentation time and temperature. So for an enzymatic fragmentation workflow to really be considered easy to use, it has to be both tunable and consistent. So here we're highlighting the tunability on the left, where we're modulating the library size using fragmentation time and temperature with consistent results between the replicates. And then on the right, we're highlighting the robust performance with inputs that range from 500 nanograms to 100 picograms. So with identical frag conditions, we're able to achieve nearly identical library sizes across this 5,000-fold range. And as previously mentioned, the generation of sequence artifacts is also a problem that plagues the existing enzymatic fragmentation workflows. So chimeric reads are one such artifact that can be inflated up to 10-fold in comparison to sonication. So not only does the overabundance of chimeric reads make it difficult to determine which are true variants in your sample and which are artifactually generated as a part of the library prep process, but it can also significantly reduce your sequencing economy. So our kit virtually eliminates the excess chimeric reads relative to sonicated samples, and it reduces that rate relative to solutions that are available from other vendors. So to summarize, our Watchmaker DNA Library Prep Kit with fragmentation harnesses the scalability benefits of enzymatic frag while not compromising on sequencing accuracy and ease of use, really challenging the idea that sonication still remains the gold standard for DNA library prep workflows. And this here is my last slide. So I'd just like to point out that we do have a number of NGS and precision enzyme solutions available outside of what was shown today. So please feel free to visit our website to learn more about our products after this presentation. And if you have any questions that we're not able to address, you can also reach out to our technical team at support@watchmakergenomics.com. So thank you all for your attention. And with that, I'd like to hand it over to Melissa Netwall to present on behalf of SBT Lab Tech.
Thank you, Eleanor. Hi, everyone. My name is Melissa. I am a field application scientist on the West Coast. And I would like to introduce you to SBT's newest liquid handler, the Firefly. Oh, I lost my screen. There we go. Okay. So, SBT has been in the liquid handling space for over 20 years now. Just to give you a brief introduction of some of the other instruments that they have. The first instruments that were released were the dragonfly and the mosquito. Let me pull up my little marker over here. There we go. All right, so the first ones were the dragonfly and the mosquito, both of which utilize true positive displacement liquid handling to enable precise, accurate, and dynamic capabilities. In recent years, however, SBT has expanded its portfolio through key acquisitions, one of which was Apricot, who has also been in the liquid handling space for over 20 years, but utilizes air displacement rather than true positive displacement. So what SBT has done is they've combined these two technologies into one instrument, which has the Apricot's dual core head, dual core head, capable of picking up 384, 96, and strip tips without changing out the head, along with the dragonfly dispense head, which has the ability to dispense anywhere from 4 mils down to 200 nanoliters without changing any of the liquid class settings. All right. In addition to the dual core and the dragonfly dispense head, it has 16 deck positions, a gripper which enables fully automated workflows and options to add an orbital shaker or plate thermal module to the basement below the deck, right here. All right. Another thing here, and I think, apologies. I think Krystal mentioned this, but it also fits on any standard benchtop. We utilize the vertical space, and so it's a really approachable instrument for folks who are just getting started or potentially have a smaller lab space. All right, and just to review in a bit more detail, the pipetting head, again, is a 384 well head capable of picking up 96 tips or strips of 8 or 16. This enables the head This is enabled by the head interacting with universal tip cassettes rather than 384 or 96 individual mandrels interacting with 384 or 96 individual tips. There are two advantages here. The first being able to seamlessly go between tip types without having to change out the core of the head itself. And the second is the lack of that air differential across those different channels results in super tight CVs across your plate. Moving on to the dispense head. This head is based on SPT LabTech's Dragonfly dispense head. This works through, again, that true positive displacement. So rather than air being the force in contact with the liquid, there's a plunger within the syringe in direct contact with your liquid. This enables a dynamic dispense range of 4 mils, which is the entire capacity of the syringe, down to 200 nanoliters. And because the dispense head is liquid class agnostic, and because the air is not the primary force involved in the liquid handling, the dispense head is liquid class agnostic, meaning you're able to aspirate dispense anywhere between 70% alcohol and 70% glycerol without changing any of the liquid class settings. Another huge benefit of the Firefly is the dead volume. Rather than a full SBS footprint reservoir for reagents, you are able to utilize our standard and low dead volume reservoirs to reduce the volumes that you're utilizing from these kits. And so those dead volumes are 200 microliters and 70 microliters respectively. In addition to this, the dispense head, like Krystal said earlier, is rather fast. For example, if you're utilizing 3 channels, you're able to fill a 3 to 4 well plate with 5 microliters of liquid in less than 1 minute. All right. So the Firefly is a chemistry agnostic platform. And what we mean to say here is that we have developed methods ready to go for multiple different sequencing kits, including Watchmaker. Another really great thing about the Firefly is the software. So the software enables users without a coding background to seamlessly write and execute protocols while still maintaining a great deal of control over the liquid handling properties. One of the great examples that I like to give is, rather than programming each step of a tip touch, we just have a drop down within the properties that you can select. And you can also easily select aspiration and dispense heights. If you're ever curious to see a demo of the software, we have a lot of really great resources on our website, but you can also feel free to reach out directly. Happy to walk through it with anyone. And another thing, like Krystal said, we're always looking for more ways to engage with our current customer base for method development and gathering feedback to continue improving our Firefly platform. It has been really fun to work with Krystal and her team, along with Eleanor, in getting their Watchmaker protocol up and running on the Firefly. And with that, I would just like to thank Krystal and Eleanor for their time today. And her team at TGen as well. They're also a really great group. And please feel free to reach out if you have any questions. And with that, I'll turn it back over to Kira.
Thank you, Melissa. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. And we'd also like to ask attendees to take a moment after the webinar's ended to take our exit survey and to provide us with your feedback. And so now we'll start with the Q&A portion of the webinar. And the first question for Krystal is just which public health agencies are using your Group A strep data?
Hi, Valentine. I see you out there asking questions. I used to work with him and love him to death. So sadly, we only are sharing our public health data locally. So it's just within the state and some counties and public health entities within Arizona right now. So it doesn't go beyond that.
Okay. And another question for you is, are there any updates on what this IGAS genomic program has developed into currently within the EZ surveillance project generally? Is it being considered by other health departments to help shape their infectious disease surveillance programs?
Yeah, I think we are the only, according to our epidemiologist, I think we are the only one right now that's doing a state genomic surveillance program for this organism. But I have seen some literature out there. I think there was one for like Idaho or Indiana talking about the need for it after, I think just after COVID-19 in 2022, there was a rise of pediatric cases that kind of led some folks to reconsider whether they should be doing surveillance on this. And I believe it's North Carolina or South Carolina, South Carolina has been in contact with our epidemiologists trying to collaborate on optimizing a workflow there as well.
Okay. And I think this one is for Melissa. You mentioned that the dispense head is liquid class agnostic. How does it work with different liquid viscosities typical to an NGS workflow?
Yeah, so like I said, master mixes are the typical, one of the typical reagents used in NGS workflows, along with some like extra SDS-like reagents. And so the way that it works, like I talked about before, is that true positive displacement. So rather than air being in direct contact with your liquid, it's going to be that plunger within the syringe. And so the way that enables us to remain liquid class agnostic within certain parameters. We do have to tweak things every once in a while, but it is because of that true positive displacement.
And I think this next one is for Eleanor. Are there any solutions for Watchmaker RNA library prep kit with Polaris depletion?
Yes, there are. So we're talking about just general automated solutions. If we're talking specific on the Firefly, we have not yet developed a method for the Firefly, but if anyone is interested, I'm certain SPT and Watchmaker would be willing to partner to make that happen. On other automated systems, we have a number of projects that are kind of outstanding. If you want to learn more about those, please feel free to visit the Watchmaker website.
And Krystal, what are the typical sample throughputs that your lab is currently experiencing on a weekly basis? Do you wait and consolidate when you have sufficient samples before performing a run? How do you time that out?
Yeah, so we have several different projects going on, but for group A strep, we probably only get like 100 samples per month. And so we typically will wait until we have at least like a half of a plate, maybe like 48. And so we do have time constraints in terms of extracting from the plates. So we still continue with extracting DNA, and then we might just plate that and QC and normalize all of it at once to at least have like 48 samples, because IGAS is a pretty small organism too. So that kind of plays a role in whether you can actually sequence it, even if you do finish prepping it. So we usually wait for about 40 or so samples before we go through with the prep. But we do have a lot of we have like COVID samples and stuff like that too, where sometimes we're getting hundreds per week or even thousands. So then everything goes really quickly when we're doing stuff like that.
Okay. Let's see. So someone had another time question, but it's a little bit of a different time question. What were kind of like the ballpark timeframes, or like weeks or months, for different milestones that you covered today in your transition to the automated prep? About how long did like those different steps take?
For the automated prep, I think it was a pretty quick delivery, and it was a new instrument, so it took a little bit to get it installed properly and make sure we had everything good going there, but not very long at all. I would say in terms of like I think we started doing applications training in March. We had a protocol developed like a week later that we tested out, and we've been optimizing that. And I think that was pretty much ready to roll in May. And that's with us not doing it every day or anything. We're just kind of doing it on the side. So maybe spending 2 days a week or something playing with it.
Okay. Let's see. And for Melissa, with positive displacement, how does the instrument deal with potential cross-contamination issues?
Sure, this is something I probably did not voiceover as much as I should have, is the Dragonfly dispense head is a non-contact dispenser. And so it hovers over the plate, and like Krystal said earlier, the deck moves underneath it. We have not seen evidence of cross-contamination utilizing either the Dragonfly or the dispense head within the Firefly. Like I said, the Dragonfly has been around for about 20 years. So that's one of our favorite instruments at SP2 LabTech.
And what's about the typical time to process 96 samples with Watchmaker LP on Firefly? I'm not sure who that, I think that's kind of like a group question.
I think for the 24 reaction protocol, it's 1.5 hours, not including thermal cyclic times. So I don't know, maybe Melissa, if you want to talk about how much extra time, I would guess it's not much more. We haven't done the 96 yet.
And automation in general should only make things faster. So what the kit says, I think it's 130 for a timeline. I would imagine typically that is sped up by utilizing automation. It's also much easier to know what your timelines will be because you're not focused on manual prep, and that's going to vary between each individual person who does those types of workflows. Eleanor, do you want to chime in with anything? You are the automation guru here.
Yeah, I mean, generally we don't expect to see a massive increase in time, maybe a slight adjustment just depending on how long it might take the instrument to be able to distribute reagents across that 96 wells. But with the positive displacement head, I know that can happen really quickly and fairly efficiently. So I don't think we're talking about too much additional time. I'd probably anticipate between like 20 to 30 minutes, maybe max, to scale up to that 96 reactions. But hopefully, Krystal, you'll know more soon if you guys plan on scaling up.
Yeah, I think you're right with that. Especially, I think the only time-consuming part is probably just bead cleanups, because you have to mix it slowly. But you have a whole head, so it's not like it wouldn't change it. Never mind, take that back.
Exactly.
I would imagine that it would go faster because, like a manual prep, you're typically using a multi-channel, so you're doing one column or row at a time, whereas you have the entire 96 well head. I have not done a bead cleanup by hand in a very long time, so I don't remember the timing of that. But I don't recall it being a good time.
It's way faster when you automate.
Gotcha. Oh, and when validating your methodology, do you consider various sample types, whole blood versus buccal swabs versus dry blood spot cards, and so on?
Yeah, I think
sorry, they didn't specify a person.
For us, I think we would, depending on the project type. So we might review and re-optimize something based on that project sample type. So yeah, I do think that is really important, especially if you have like FFPE or some other kind of degraded sample type. But we we tested our kits with like a cell culture known genome, so that we would know how well it was performing, and we knew what mutations it had and didn't have. So that's how we typically test whole genome kits, at least. But if you're after that, then yeah, you would definitely try some more complex, difficult to prep samples.
This one's for Eleanor. How long does it usually take, and what is required for the automation team to establish a protocol? And they're specifically looking at RNA prep here.
Yeah, that's a really good question. So generally, it's going to be dependent on the instrument that's being used, and also how much kind of expertise the end user has as well. So if we just look at any given project that our team takes on, I'd probably say for an RNA prep, which is probably our more differentiated kit available on the market today, as in there are not many other vendors with workflows that are similar to our kit. I'd say it probably takes, anywhere from 3 to 6 months to develop a fully kind of verified, well-baked, liquid optimized automated solution that can be dropped onto a liquid handler. Now, this is talking very kind of generally. If we have a method that's similar that we can start with on a specific platform, then that tends to shorten down the development time. So it really does depend kind of on the customer, the specific platform, and then also what workflow we're talking about. But for RNA, it is a little bit longer of a project because our kit is so differentiated. If you have questions about a specific platform, again, if you go to our website, you can put in like a technical inquiry and we can get back to you with details on that specific platform.
Okay. And this one is for either Krystal or Eleanor. Someone's just wondering if you're doing any work on miniaturizing the preps, doing like half reactions or things of that nature.
I would love to. Of course, because that would save us money, right? So definitely interested in that. And you know, if I had the money right now, I'd probably look into a mosquito for something like that from SBT Lab Tech as well.
On that note, little Firefly pitch here. So I had a group that purchased a Firefly and they had a mosquito and dragonfly combo. And we were able to transfer their 10x miniaturized workflow to the Firefly, and the data is beautiful. So totally doable on the Firefly.
Yeah, cool. I don't know how Eleanor feels about that, but
no, it's great.
Like
I mean, in fairness, we're definitely in support of what our customers are doing. So if that's kind of the next step for you guys, then it makes sense. And with the Firefly's capabilities, I think it makes sense that would be a platform of choice.
And a question for Krystal is, what instruments do you use for genomic DNA extraction?
I do not currently automate genomic DNA extraction for this workflow, but we do for COVID RNA extraction. We will use the, oh my gosh, what is it called? Kingfisher is what we're using right now. And our collaborative sequencing center has some other instrumentation. I think they have a Chemagic or something or other, another sort of bead-based extraction instrument. But yeah, we use column-based extractions for these.
What is the largest pipetting volume that the Firefly can aspirate or dispense?
Sure. So we have filtered and non-filtered tips. For the non-filtered tips, we have a 125 microliter tip or a 50 microliter tip. Then with the filters, that is decreased down to 100 microliters and 35 microliters respectively.
Let's see. Sorry, we're jumping around now in the sort of the topics here. Since RNA degrades very quickly, what's the recovery rate with RNA samples with regard to preparation? That might be for you, Krystal.
Well, it might be, I'll kick it over to Eleanor just after I say that for the group A strep workflow, we are extracting DNA bacteria, so not working in this particular one, but it is a good point that it degrades fast, not my favorite. Eleanor.
Yeah, so there are ways that we can kind of minimize the impact, right? Especially with RNA workflows. Generally, what I would recommend for automating an RNA workflow would be to use an actively cooled position, something that can keep your sample plate somewhere around 4 degrees, at least less than 10 degrees Celsius. In addition to that, keeping reagents also actively cooled. And then for the incubations, actually doing those in enclosed kind of temperature controlled devices. So thermocyclers or on-deck thermocyclers. Those are kind of great ways, tools in which we can use to help prevent degradation. But with the kind of quick and fast and efficient workflow that we offer, that's only 4.5 hours, there's really not a lot of opportunity for your RNA sample to degrade just due to sitting around because it happens so quickly. So it's definitely worth looking into our RNA library prep solution if that's something that you're interested in.
And another question for you. Does Watchmaker have kits with incorporated human genome depletion steps?
Yes. So in terms of depletion, we do have the Polaris depletion kit for our RNA workflow, but that's specific to RNA library prep. If we're talking about DNA library prep, we don't have depletion that's specific for a DNA workflow. So if you're trying to get rid of like high molecular weight material, let's say if you were doing like a cell-free DNA workflow or something of that nature, our kits do not include that step.
Okay. Well, that's all the time we have for today. We'd like to thank our speakers, Krystal Sheridan, Eleanor Matrullo, and Melissa Netwall, and our sponsor, SPT Lab Tech. If we didn't have time to get to your question, we'll try to follow up with our experts. And as a reminder, please look out for the survey after you log out to provide us with your feedback. If you missed any part of this webinar or would like to listen to it again, an archived version will be emailed to all attendees. Thank you for joining us for this genome webinar.
Thank you.